All right. Any questions about that process before we move on? I know it's a lot. But anything you're like just totally lost on? Okay. Here we go. So this is actually a pretty short PowerPoint, so we'll easily get through it today. I do have a video in here as well, so multiple videos today. Okay, genetic mutations. Like I said, that process usually works really well. There is plenty of room for errors to occur with that coding, the matching up, all of that. So that's what we're going to talk about here, kind of what happens when things get a little bit off. So what is a mutation? Everyone probably has a decent idea of what a mutation is. The definition is just a change in genetic material. So mutations typically get this kind of bad rap, but some of the changes have no effect. So there's mutations that occur all the time where we don't actually see any effect. I'll talk about those. And then sometimes they're actually beneficial. So it can be a beneficial mutation. But all a mutation is when the genetic material compared to the parent material is different. They are passed on to offspring because they're in the genes, right? So they end up in the gametes of the offspring. And the term for that is heritable. I think I've used that. We used that when I was talking about the Mendelian genetics and Punnett squares and all of that. Heritable just means it can be inherited. So if a parent's genome has some kind of mutation in it, it will get passed on. If it makes it into the gamete that eventually becomes a little human or a little whatever organism you're talking about, that offspring will have the mutation as well. And they can be caused by kind of a couple different main categories. So it could be a random mistake in one of those processes we just covered, DNA replication, transcription, or translation. There's a lot of code reading that has to happen throughout all of that. If errors occur in any of that, then a mutation will likely result. Environmental factors can also cause mutations. We're not really going to get into that, but like UV radiation can cause mutations. Certain toxins can cause mutations, but we're really just focusing on the sort of basic genetic mutations. I'm going to go through a few different categories here of mutations. Point mutations are where a single nitrogenous base is altered. So it's at one point in, the DNA or the RNA it's made. Just 1 point, one single base, an A, a T, a C, a G, a U, whatever, is altered. It's also called a base substitution. So one of those letters is substituted for another. That's a point mutation. So these are really tiny. It's not like a whole chunk of chromosome is mutated. It's just one base. As I mentioned, some mutations don't cause any change. So that's called a silent mutation. Silent as in you don't hear it or, I guess, see the results of it in this case. And by substituting one base for another in a silent mutation, the same amino acid is still inserted. So like I talked about on Monday and you just saw on that video, there are multiple codons that can code for the same amino acid. So there's some overlap. So if it so happens that one base is substituted for another in one of those codes, but it still codes for the same amino acid to be brought in, there's no overall effect. That is a silent mutation. So really no effect on the protein at all. A missense mutation does change the amino acid. So this is kind of the other option. So silent or missense, there's one more that I'll talk about in a second, but a missense mutation, one base is altered, and then that leads to a code for a new different amino acid than originally would have been coded for. That might have an effect on the protein, that might not. It will have an effect on the amino acid sequence because you have a different amino acid in there. The functionality of the protein will likely be affected, but may not. And then you have what's called a nonsense mutation, which is where the codon, the original codon would have coded for an amino acid to be brought in, but now one of those bases is changed and it changes it to one of those stop codons. So there's those 3 stop codons. So when a codon is changed from a normal amino acid coding codon to a stop codon, that's a nonsense. So silent, missense, and nonsense. Those are the 3 kinds of point mutations. So this is kind of what it looks like in a diagram form. So the original here is the original codon that we have is on the left here. So mRNA would be AAG based off of the DNA level. The DNA TTC codon, or 3 letters, I guess it's not a codon in DNA. That's going to code for this amino acid, lysine. A silent mutation in the DNA, if that C is changed to a T. So here we have a T instead of a C. And then it ends up changing the mRNA codon, but we still get the same amino acid, so it's silent. Yes, there was a change in the base, but the amino acid turns out the same, so we don't really see any effect from it. Nonsense, we have a change to a stop codon. So at the DNA level, instead of TTC, we have ATC. And then in the mRNA strand, that's going to be UAG, which is a stop codon. So UAG here. So instead of being a code for this lysine, it's a code to stop the whole process, which can cause some major issues, probably a totally non-functional protein, depending on where it is in the process. And then missense, that's just where you have a change in one of the bases and you get a different amino acid. So it's not a stop codon, it's not that serious. I guess it could still have a pretty serious effect. But arginine here is coded for instead of glycine. So different amino acid just from one change, a change in one of the bases. It just depends on what that base change is a lot of times. Okay, so those are the 3 types of point mutations I want you to be familiar with and kind of be able to identify if I give you an example of one of them. And I want to stop here and talk about a disease that is caused by a single point mutation, which is sickle cell anemia. Has everyone heard of sickle cell anemia? Okay. So this is a somewhat rare disease. Basically, I'll talk about the genetics behind it, but it's a recessive trait. So each parent has to carry the allele and the offspring, the child has to have both of those recessive alleles in order to have sickle cell anemia or sickle cell disease. Parents can be carriers if they're heterozygous, but doesn't show symptoms. So that kind of takes us back to what we were talking about with the genetics chapter. So what happens, typically red blood cells are these kind of donut-shaped, round indented in the middle cells here. In sickle cell anemia, a lot of the red blood cells end up in this sickle shape, and they don't carry oxygen as well. So red blood cells are carrying oxygen around. In the sickle shape, they don't carry oxygen as effectively, and then they also can clog arteries and veins. They don't move through capillaries or blood vessels quite as well, which causes anemia. So that's basically why we get the term sickle cell anemia. Okay, so what happens? There is not much, there's not much change in the gene that causes this in order for sickle cell anemia to be the product. So what happens, this is a gene called HBB. It has to do with hemoglobin. So in the non-mutated version, so the normal HBB sequence, there's an adenine here, which is going to code for glutamine. I think that's the amino acid. In the sickle cell or abnormal HBB sequence, instead of that A, you have a T. So a T gets inserted instead of an A. That codes for a different amino acid, valine. That's all that causes sickle cell anemia, but the effects are wide-ranging in a person's body, even though there's just the one base pair substitution. So point mutation causes sickle cell anemia. So would this be silent, nonsense, or missense mutation? Missense, yeah, it's coding for a different amino acid. So it's a missense. It's definitely not silent, and the nonsense codes for a stop codon. So this is a missense point mutation. I have a video, so it's easy to kind of forget that a lot of what we're talking about, especially with these genetic mutations, has really major effects on people. So I want to kind of bring in a personal story about a girl who has sickle cell anemia, so you can see a little bit more about the disease and kind of learn more about some of the symptoms of it. You're not going to be responsible for any of this, but it's more kind of just for your information. And like I said, you can break it down to a point substitution, like a very small mutation, but it's important not to forget that it affects people in very large ways.
When other kids used to go outside and play, I would try to do that. Then I come home and very sick and have to be back in the hospital.
17-year-old Alexandria Young of St. Louis, Missouri, has sickle cell anemia, among 72,000 Americans with the disease.
Among all Americans, approximately 1 in 1,500 to 1 in 2,000 newborns will be diagnosed with sickle cell disease.
In people with sickle cell disease, their red blood cells become deformed, turning sickle-shaped. The bone marrow tries to make more red cells to make up for the loss, but can't keep up, causing anemia. Their new shape keeps them from moving properly through the body, and the misshapen cells can jam up and stick to the walls of the blood vessels. These clumps cut off oxygen to healthy tissue, delaying a child's normal growth and causing fatigue and extreme pain.
It's like a hammer beating at you like for a long time.
I never heard anybody scream that high and just wail out, and we were like, what's wrong? And man, she had had it so bad where her eyes just roll back up, just roll back and just in pain, just screaming, yelling, and we didn't know what to do.
And I thought she was just talking in her sleep or something, but she had mentioned that she was ready to go home. And I said, Alexandria, you are home. We're home now. She said, no, Mommy, I want to go to heaven. That's my home. And
Okay, deep breath.
Dr. Michael DuBon has been Alexandria's hematologist since she was 4 years old.
Any headaches, double vision?
Fatigue and pain are not the only issue in sickle cell disease. Essentially, every organ in the body can be damaged by the lack of oxygen. The kidneys, the heart, the eyes, and now we know even the brain.
In addition to pain, probably the most feared complication is that of a stroke.
Yeah, I'm right here as a silent stroke.
When Alexandria was 12 years old, a brain scan revealed that she had indeed had a stroke.
I was very scared because I thought only older people would have a stroke and not 12-year-olds.
Silent strokes like Alexandria's were not even recognized until a small research study in the 90s was conducted to better understand sickle cell disease in children. MRI showed that strokes occurred when the misshapen cells got stuck in the brain and cut off oxygen to it.
Certain locations of the brain control your movement. They control your motor ability, okay? There are other areas of the brain that if we removed it, it wouldn't alter your movement at all. It would alter your ability to think. The area of the brain in the frontal lobe is the area of the brain that's more likely to have silent strokes. So most of the children with silent strokes, they're actually walking around, interacting with their family in a fashion that makes people think that, yeah, this child is doing well.
Dr. DeBoin has Alexandria on a research protocol, hoping to stop the strokes. Monthly blood transfusions, called apheresis, remove Alexandria's damaged red sickle cells and replace them with red blood cells from a healthy donor. The hope is that the healthy new red blood cells will prevent her from having future strokes. Dr. DeBoin monitors Alexandria's condition closely, including annual brain scans to look for further damage.
I pray constantly, about Alexandria, that she hasn't had another silent stroke.
The young family won't know the results until their next visit to Dr. DeBoin.
Out of the clinic. 3 days.
Staying on this protocol, it has dramatically improved her classroom performance, dramatically just improved everything in her life is just completely turned around.
Whew, it's just like a switch, man. It just totally changed her. She started to grow.
Congratulations, that's fantastic. Straight A's.
How was her test results?
We had the MRI for brain performed a couple of days ago.
It's to make sure that she hasn't had any new episodes of silent strokes.
That's right. And the answer is no. There's been no evidence of progression. So it's all done and it's really good news. Okay.
All right, so yeah, like I said, just a reminder, I talk about all of this stuff from a basic bio perspective, but especially when we're talking about genetics and genetic mutations. Oh, shoot. Oh yeah, okay. It's important to remember there are some, major repercussions from even just one base being substituted on one gene. It's pretty amazing. And if you're interested at all in any kind of medical research, there are so many different, well, not only genetic diseases, obviously, but we're talking about genetic diseases. So many kinds of genetic diseases where they're making huge strides, like with sickle cell anemia, trying to figure out how to allow a patient to live, a relatively normal life. And actually, we're going to get into biotechnology and genome stuff next week. And they're actually trying to figure out how to change genes in living people. So potentially you could fix this issue and then that person doesn't have sickle cell anymore, which is pretty amazing. So that's a huge, huge new development that we'll talk about. Okay, so sickle cell anemia, what I really want you to know for this class is it's a missense point mutation in a very specific gene. But yeah, anyway, hopefully that was somewhat interesting and eye-opening to you. All right, next we have not just point mutations, there are more significant mutations called frame shift mutations. And what these do is essentially shift the entire reading frame of the during the translation process. So what does that mean? Essentially, it's just an addition or deletion of a base or multiple bases. So if that code is read by 3 letters at a time, if you add a base in there or take one out, that shifts everything that's being read after that addition or subtraction, right? It just shifts everything and changes everything after that addition or deletion. So what's being shown here is we have these 3 U's. And then one of them down here on the frame shift mutation, we're deleting one of those U's. So before, the 3 U's coded for PHE, whatever amino acid that is. Instead of 3 U's, now you have a UUG that's being read, and that codes for leucine. And then everything after that is now changed as well, because you totally took out one of them and everything else shifted. So now the codons are all completely different after that addition or deletion. Yep.
So 3 of the same letters can't really be like
Can't be what?
like the 3 you
Yeah.
they can't. One was deleted because 3 can't be together, right?
Oh no, they can be. So UUU will code for an amino acid. That's totally fine. But the mutation is that one of these is deleted. So for some reason, something random happens and that U was just taken out. So now instead of UUU, you have UUG. And then now, because we don't have that G shifted over, and now the next 3 are different as well. So rather than reading that GGC, it's now all shifted over by 1. Yeah, good question. So the mutation is that addition or deletion. The result is that then everything else is read differently because the grouping will be different after that. So yeah, this is going to have much larger consequences typically than a point mutation because it shifts the entire reading frame on that mRNA. So you can see in this example, it arrives, so this is the normal gene, and there were a lot, multiple amino acids strung together here. Instead of continuing that, not only do we have different amino acids at these 2 points, we also arrived at stop codon well before we got to the end of what should have been the amino acid chain. That doesn't happen all the time. It just depends on what the change is. But yeah, really significant mutations with frame shift mutations. This can result in really debilitating genetic diseases like cystic fibrosis. Cystic fibrosis involves a buildup of mucus in a lot of different organs, but in particular in the lungs. Tay-Sachs disease, I talked about that a long time ago when we were talking about lysosomes. That's usually fatal to children before they're 5, but the lysosomes don't function correctly. And then also a much more common one, genetic high cholesterol. So a lot of people, even if you eat really well and you have a healthy diet and you exercise, some people still have high cholesterol because of basically a frame shift mutation. So if you hear of someone who has genetically high cholesterol, not from poor diet or lack of exercise, but just from a genetic standpoint, it's likely due to a frame shift mutation. So I guess that's not a debilitating genetic disease, but the other 2 definitely are. All right. What else do we have? Okay, so more large scale mutations. Chromosomal mutations. I've talked about these already, actually. So this is hopefully a review. This is when huge chunks of a chromosome are changed. So we're not just talking about bases shifting a little bit like the individual letters. Now we're taking multiple genes or multiple sections out of a chromosome if it's a deletion. Inversions will just flip the order. So instead of A, B, C, D, it's A, D, C, B. So an inversion. hopefully these sound kind of familiar. Duplication, where a segment is duplicated, so you have two of the same ABCD. Basically here, BCD is repeated twice when it shouldn't be. That's a duplication. And then translocation. I don't think I used the reciprocal word before, but translocation is where one segment of a chromosome moves to an entirely different chromosome. So those typically have fairly large consequences as well, as you might imagine. If you're changing massive amounts of DNA at once, you're going to have a lot larger, much larger effect than just changing one point mutation or one base pair. All right. And then to kind of wrap up, mutations and evolution. So mutations, like I said, are not always a bad thing. They're actually the starting point for any kind of evolution. So you have to have some kind of change in the genetic material to allow for evolution. Beneficial mutations will be more likely to be passed on. So mutations can be a good thing. However, if a species changes too much too quickly, like a whole bunch of mutations, that can be pretty harmful and dangerous. So fewer mutations are typically going to be better for a species, but in order to adapt to new environments, mutations are going to be necessary a lot of times in order to allow for that to happen and allow for that to be passed on to the offspring. So yeah, a balance, like I said here, a balance between new variation and kind of status quo will help increase the health of the species overall. So we're talking overall species, not just individuals here, since we're talking about evolution, which we won't really get into evolution in this class in any detail. And then some interesting info about human mutation rates. So we can actually directly measure how quickly our genomes, which is just all of our DNA, is mutating. And this is kind of, when I read this, it was kind of shocking. For every birth, there's about 70 new single bases or nucleotides that vary compared to the parents. So you guys probably have at least 70 different nucleotides in your sequences compared to your parents. And then your offspring will have 70 new single nucleotide variations as well. So it's pretty amazing how many changes actually occur. Again, a lot of them, you don't really see much of an effect. And I meant to look into this. I don't know why this is the case, but 76% of the mutations come from the father. So from the paternal genome is where they're tracing a lot of these mutations back to. And I have no idea why. Now I want to go look it up. I have no idea. But as the father ages, typically that rate increases as well. So older fathers, if they're having children at an older age, will be more likely to have children with even more mutations in their DNA. So yeah, we can directly measure how much our species is kind of mutating now, which is cool. Okay, and that is all of that chapter. So like I said, pretty short lecture on that. Don't forget about the quiz Friday. It'll cover through this material, so through the genetic mutation lecture. And then I'll see you guys again on Monday. So reminder, no class Friday. Take the quiz, that's all you have to do, and I'll see you on Monday.